So we gave the basic overview of cytoplasmic male sterility, and there's a conflict of interest. And then as Brian foreshadowed, there's a certain set of common characteristics of cytoplasmic male sterility when you look in natural populations, and that is that you typically see multiple mitochondrial genotypes, and each mitochondrial genotype can cause male sterility by a different mechanism or somehow genetically distinct. And we know that the mitochondria are genetically distinct because the nuclear restorers tend to be specific for individual mitochondria. So if we have some nuclear restorer genes that can restore pollen fertility for a particular mitochondrial type, those restorer genes will not be effective against a different mitochondrial type. So we have our multiple mitochondrial types, and each mitochondrial type will typically have its own set of specific restorers. Okay. And if you think about it, it's actually a fairly complicated problem to figure all of this out, because how can you tell if the mitochondria of two different plants are different? Well, in order to do that, you have to have restorers that are different. You have to be able to put the mitochondria into different nuclear backgrounds and show that they respond differently in different nuclear backgrounds. So in order to distinguish 5 mitochondrial types, you have to have 5 genetically distinct nuclear backgrounds that contain 5 different sets of restorer genes. And the same thing is true for the restorer genes. How can you tell that different restorer genes are distinct? Well, you can only tell if you have appropriately different cytoplasms. So actually working this out is extremely complicated because you need reciprocal polymorphisms in order to see the other side. And this is generally true of arms race polymorphism situations. And it's even harder than it sounds, because in order to tell the two mitochondria are different, you have to guess that they're different and put them into different nuclear backgrounds before you know that they're different, right? Because if you don't know that they're different, then how do you know to put them into different nuclear backgrounds to see how they respond differently? So you have to do this huge number of crosses in order to see these things without really knowing what you're looking for. So it's a very complex situation. But in spite of that, people have worked out that there's quite a bit of polymorphism, and modern genetic tools give you some clues, and so it's made it a lot easier. But anyway, it's characteristic of these species that there's different mitochondrial types, different sets of nuclear restorer genes, and that in natural populations, there's mixtures of mitochondrial types and nuclear restorer genes. Now, the nuclear restorer genes are also further complicated, because if the nuclear restorer genes were at a frequency of 100% in the population, you could not see them. If all nuclear backgrounds have the same restorer genes at a particular locus, then in some sense there's no way to know that they're there, because you can only tell when something's there when you see variation. So there may be a variety of nuclear restorer genes that actually are present and are at a frequency of 100%. We don't see those. We only see the ones that are polymorphic. So what we tend to see in natural populations is significant polymorphism for the different mitochondria, and then we see multiple loci, multiple genes for restorers that are themselves polymorphic. And then one of the characteristics is that the polymorphisms tend to vary over space. So if you look in one population, you might see a particular mitochondrial type is very common. In another population, another mitochondrial type will be very common. In one population, some of the restorer genes might be at high frequency, and in another population, the restorer genes will be at low frequency. So you're seeing this incredible diversity over space and over time in the frequencies of these genes, which is consistent with this notion that there's some sort of conflict or arms race going on, and we tend to see this. And so I think I'll show you, I'm not going to show very many slides today, but I'll show you just one set of slides to show you the kind of variation that we see. So I'm going to share, do that. So this is an example of spatial variation. Let's see if I can. There we go. Spatial variation. On the bottom is a species that we'll talk about a bit called Plantago Lancioleta. If you happen to be from the northeastern part of the United States, you know this plant, even though you might not think you know it. It's in every lawn as a little weed. It's a very short little plant. And you can see that among different populations, the frequency of females, some populations have just a few percent females or male sterile plants. Some have 10 to 20%. Some have as many as 30%. So you see this variation across space in the frequency of male sterile plants. Now in Thymus species in France, there's tremendous variation. So you can see these different species vary. Some of the species, the frequencies are often above 50%. That's the one in black here, Masticina. In Zygus, you see the frequencies vary. They're lower, but they're still very high. And there's huge variation between populations. Now this was a study done in 1986, but I still think it's to me, the most fascinating study of cytoplasmic male sterility, because to me it really captures what I think personally is going on in a lot of these situations. So this is a single field. These plants are very short, so they're just a couple of inches high. And this is 50 meters. So the field's about 225 meters by about 350 meters. So it's a sizable field, but not huge. And in this whole field as a whole, if you averaged over the whole field, there were a lot of plants. I can't remember the exact number. 94% of them had one mitochondrial type, and 6% had another mitochondrial type. So it was very high frequency for that one mitochondrial type. And the restorer genes for that common mitochondrial type were also very frequent. So most plants in this population were restored hermaphrodites. They had full pollen. But there were, among the plants that had the rarer mitochondrial type in 6%, actually in a couple of locations, P1, P2, those are population 1, population 2, 3, and 4. In those locations, the rare male sterile cytoplasm, mitochondrion, was very common in these locations at much higher frequency. And the restorer genes for this other mitochondrial type were rare in the population as a whole, consistent with the fact that the mitochondrial type was rare. But in these locations, the restorer genes were in higher frequency. And so the interpretation is that the population had mostly one mitochondrial type, and then had restorers for that mitochondrial type. So it was mostly a hermaphroditic population. But that a cytoplasmic or mitochondrial type migrated in and landed by a long distance migration into this population, carrying another mitochondrial type which caused male sterility, for which the restorers were locally rare. Now those plants are then male sterile. They produce more seeds than the hermaphrodites do, so they tend to spread locally. These plants disperse over very short distances. They're very small plants. The seeds move probably less than a foot or two in any year, in any generation. So what you get is a little local patch where the male sterile plants begin to spread because they make more seeds. That sets up very intense selective pressure for the restorer genes for those particular cytoplasms to come in, because as we've talked about, in this little patch, pollen becomes rare because you've got a lot of male sterile plants. So any plant that can produce pollen around a bunch of plants that are male sterile has a lot of fitness through its pollen as a father, so to speak, because there's very little competition with other plants that produce pollen. So the restorer genes spread very quickly. So this is what we're seeing a little focus of male sterile plants spreading, and then the restorers coming in and the restorers increasing in frequency very rapidly because of the frequency dependence. And this same process seems to have happened in 4 different spatial locations in this population. And so that gives a sense of what the dynamics of this situation might be in natural populations. Now, eventually, the time scale can vary, but in some cases, maybe not in this species, but in other species, a male sterile mitochondria might spread through a population very fast and drive out the other mitochondrial types and cause a complete turnover. That would then favor the restorer genes to change very much locally. And then if you imagine a whole bunch of populations doing that, then you get a very complex spatial mosaic with colonizations and extinctions being driven by this male sterility phenotype through this process. So you sort of like a little outbreaks of infection epidemics happening across time and space. And that's my view of how this male sterility actually probably happens. This is a little bit of an abstract argument, and I'll just make it very quickly. But my argument is that if you only have a couple of different mitochondrial types, then what you tend to do is get fluctuations like we just saw. That what will probably happen in that field is the type that's rare, the mitochondrial type that's rare will become common, but the other type won't go away. And so over time you'll get fluctuations back and forth between those 2 different types. But as you add more and more different mitochondrial types, the probability that a mitochondrial type will become absent from a local population increases. If a mitochondrial type is absent from a local population, then the restorers for that mitochondrial type have no benefit, and they may become absent also. Then if that mitochondrial type which is absent lands in that population, it can spread very rapidly because the restorers are absent locally. And as it spreads very rapidly, it drives out the other mitochondrial types. So my vision is that as you get more and more different mitochondrial types, then across space you have more and more of the diversity tends to increase. But in each little patch, you actually get a decrease in diversity because the more types there are, the greater the probability that one of the mitochondrial types is locally absent and can invade and spread as an invader because the restorers will be absent. And when a mitochondrial type spreads as an invader, it tends to drive out the other mitochondrial types. So what you actually get is the local diversity decreases, but the diversity across space increases as you get more and more different types. And I emphasize this because I think this is actually widely true of the various arms race types of dynamics that we see in biology with host-parasite interactions and many types of attack and defense situations that we see across all organisms, that you tend to get this general process. And this is conceptual view of what I think is going on. And there's some evidence for this in different situations, but it's a very complicated process, and we don't know that much about natural populations. Then I just want to show you a couple slides showing you the amount of diversity that actually occurs. So this is the same species that we looked at initially in this population, but done later with better molecular tools. And it turns out that there were 3 different male sterile cytoplasms plus yet another mitochondrial type which is unidentified. And this was the spatial variation in the frequencies of those types across different populations. Now, this is a pretty big spatial scale. If you look at a smaller spatial scale, as we looked at previously, you might actually see even greater variation because this species has a short dispersal distance. Now, in this case, they classified one of the mitochondrial types as a CMS2, and that's in this hatch type. But when they actually looked with better genetic tools, they actually found 4 different types that were classified as CMS2, showing that there's actually maybe even more diversity in this particular species that's hidden, because remember how hard it is to determine whether different mitochondrial genotypes are actually distinct from male sterility. You have to get the matching restorers also. Now the restorer frequencies for 2 of the mitochondria vary somewhat over space, but not greatly. And I think in this case, this is the wrong spatial scale, because a big population like the one we saw at first, we actually saw that the dynamics were actually playing out over much shorter spatial scales. And so I think actually if you looked at the proper spatial scale for the dynamics of the species, you would see a lot of variation in the restorer frequencies. And in this case, these populations are too big, and so they're averaging over many different subpopulations in which the actual dynamics occur. But anyway, that's...
Does the spread of these restorers in populations pretty much just depend on the genes for pollen dispersal?
It depends on pollen dispersal. I don't know what you mean by the
Or
genes for
are there
pollen
genes that
dispersal.
let it, like if the wind blows it, does it blow it farther if they have different shapes or something?
In the species that I was talking about, it's wind pollinated, but they're very small plants, and so the average dispersal distance, pollination distance, is very short. But occasionally there's long range pollination. In the seeds also, the seed dispersal distance is also very short, but occasionally there's a long range dispersal. So that's why I think the spatial scale in that species is on the order of a few feet. So when you look at a field that's 200 meters by 300 meters, you're really averaging over a lot of subunits on which the dynamics are playing out. So it's very important if you want to study a situation like that to match your sampling scale to the scale of the dynamics. But of course, that's a real challenge to know all of that in advance if you're actually imagining doing a field study. And these studies were done before a lot of modern genetic technology was available. So they were done with extremely labor-intensive crosses in the greenhouse to figure out what the genotypes were.
So do you know if like the frequency of the restorer genes in the population would affect selective pressures on the ability to disperse pollen?
I don't know that there's I don't know and don't see a reason why there would be a correlation between pleiotropic correlation between pollen dispersal distance and restorers.
Or if
If
they're
we
just
have
like,
time, we'll talk a little bit about molecularly what the restorers are doing. And um, there's no intrinsic reason to think that the restorers would be associated with pollen dispersal distance,
or
but
is
it's
it like
possible.
a very low frequency of the restorers in the population? Would selective pressures need to increase the ability of pollen to disperse to sort of restore the population? You know what I mean?
Yeah, but that's a species level selection argument. And so that's not how things work,
Okay,
if you know what I mean. I said it very quickly because I think you know what I mean. Do you?
somewhat.
Maybe somewhat. Well,
I'll think
think
about
about it. I mean, it's the survival of the population isn't generally going to affect the evolution of the traits. It's individual fitness or genotypic fitness that matters. So you have to really ask, how does the trait associated with the genotype affect the spread of that genotype? Anything else is going to get you going in the wrong direction. So if it's true that the population, if certain traits tend to make populations go extinct, then the consequence of that is that the population will go extinct. If that's not in the, if the individual genotypes are favoring such traits. So that's what happens in biology. Genotypic fitness can favor traits that cause extinctions, and then extinction follows.
Okay, okay. Thank you.
Okay. So where are we? So one of the interesting things so the question is, how often does this cytoplasmic male sterility happen? It's actually a hard question, as it turns out. We see mixtures of male sterile plants and hermaphrodites on the order of 1 or 2% of hermaphroditic species. So you would say it's not common, but there's a lot of flowering plants. So that's hundreds or thousands of species that have this. But here's the real kicker here, which is that when you hybridize hermaphroditic species, sometimes you see male sterile progeny. Now what's going on? Well, imagine that in one species you've got some mitochondria which can cause male sterility, but the restorer genes are at a frequency of 100%. So all the plants are restored male sterile plants, which are just hermaphrodites to your eye. And as long as you look in that one species and look at the genetics of that one species, it's just a hermaphroditic species. And there's no way to know. But if you hybridize and you take the mitochondrion from one species and you put it in a nuclear background of another species that lacks the restorer genes, then you get a male sterile hybrid. And in the extreme case, and this is extreme, but one has to wonder, it's possible that most hermaphrodites are just restored male sterile plants. So when we say that most plants are hermaphrodites and male sterility is rare, that's true, but it could also be true that most hermaphrodites, hermaphroditic species, are species in which all the plants are restored male sterile hermaphrodites. Now that sounds a bit weird, and you don't want to necessarily push that too far. But on the other hand, when you do crosses between species, you do see male sterility. And actually, one of the ways that people find new male sterile mitochondria, if they're doing breeding studies in agriculture, and we'll talk about this in a minute perhaps, is that they do crosses, they do hybridizations of the wild relatives, and that allows them to find new mitochondria. So this is a real phenomenon. It's not totally a speculative thing. And so I think that's interesting to keep in mind, because it really gives you a notion of conflict and the unit, our notion of individuals as unitary and functional and the locus of engineering design. And that can be true, and so in the hermaphrodites it is true, but it's only because the conflict has been resolved in the favor of the nucleus in this case. In other words, the nuclear genes have basically managed to dominate the conflict sufficiently that hermaphroditism is the dominant phenotype. And you can go too far that way, but you can also go not far enough. I think you have to kind of keep your mind open to that and be flexible and not forget that it exists. Because if you forget that it exists, you won't understand a lot about genotypes and how they evolved. If you go too far, then you'll be over-interpreting a variety of traits as well. So you need to be kind of down the middle on that one. And again, if we have time, we'll talk a little bit about why that's a very important perspective. And I'll just foreshadow that briefly, but I won't talk about it now, which is that meiotic drive is a situation where you have a chromosome from your mother and a chromosome from your father, and you learn Mendelian genetics. You learn that when you make a baby, that there's a 50% chance you'll transmit the chromosome from your father and a 50% chance you'll transmit the chromosome you inherited from your mother. That's Mendelian genetics, and you're taught that's how genetics works. And it is how genetics works, except occasionally there's what's called meiotic drive, where one of the chromosomes kills off the other chromosome, and so you only transmit one of the parental chromosomes and the other one loses out. And that's called segregation distortion. And that is not common, but it happens. And then the question is, when we see this 50-50 segregation, is that because meiotic drive is common, but it's repressed? And Mendelian genetics actually arises by the repression of meiotic drive. And so that's too quick to understand, but point I want to make is that this issue of male sterility and restoration is not an isolated case. Once you get this idea in mind, you can actually see that it goes to the very heart of Mendelian genetics, which is, again, we get taught Mendelian genetics is how genetics is, but actually Mendelian genetics is not logically how genetics would be. It's actually a rather surprising outcome of evolutionary history. And so just like with hermaphroditism, when you look in a population and we see all the individuals are hermaphrodites, Well, in some sense, that's surprising because maybe why aren't the mitochondria causing male sterility? Why are the nuclear genes winning the conflict so often, so to speak? And so that's an interesting question. Okay, so we'll continue on. So there's basically two broad aspects of study, and we talked about the first, which is looking at natural populations and trying to understand the frequencies of these things, the diversity, the frequency. And again, one of the really interesting things is we see multiple mitochondrial types, polymorphisms in nuclear genes, and all of this spatial variation with this turnover over time and space. So this is a very fascinating subject that really captures the essence of arms races in natural populations. And then the other big area of study is the molecular mechanisms behind all of this. And the reason why that's interesting is that you're getting a conflict between nuclear genes and mitochondrial genes. And we know that cooperation between nuclear and mitochondrial genes is essential for many aspects of genomics and normal physiological function. So we've talked about the natural history in the wild population. So let's talk a little bit about the molecular biology. Let me just look at my notes and see what I want to say. So one question is, well, how do plant mitochondria do this? And it turns out that plant mitochondria are quite weird. They're very big. They're about 10 to 20 times bigger than animal mitochondria. They have a lot of repeats. They're very complicated structures genetically. And they have a lot of DNA repeats, and they do a lot of internal recombination and rearrangement. And it turns out that plant mitochondria often have these internal rearrangements and recombinations that create new genes by rearranging the genetic material, and they form new, what are called open reading frames, which just mean new genes that make new proteins. And it's known that the male sterile consequences of the mitochondria often arise from the rearrangements that lead to new genes that somehow cause male sterility in a new way. And we don't know exactly what they're doing, but we know some of the things that they do. So one of the puzzles is mitochondria are, of course, important for all cells in a plant, but the male sterility is specific just to the pollen development or the structures that hold the pollen. And so how is that specificity occurring? And the answer is we don't know, but it seems like the mitochondrial genes are regulated in such a way that they're expressed primarily or only in the tissues that are involved in pollen development. For example, one example is known, and this is that the tissue that surrounds where the pollen grains are developed is called the tapetum. And the tapetum is very rich in mitochondria because the production of pollen is very energetically intensive and requires a lot of respiration. And so the tissue that surrounds the production of pollen, where pollen production occurs, has a lot of mitochondria. And in certain in petunia plants, actually, it's been studied where they show that the tapetal layer in a male sterile plant deteriorates, and the mitochondria basically seem to undergo a form of programmed cell death or apoptosis that destroys the tapetal layer, and so the pollen never develops. And so that's how the mitochondria can influence pollen development. And mitochondria are actually the locus, an important part of programmed cell death or apoptosis. So some of the cytoplasmic male sterility seems to be programmed cell death that's targeted to tissues involved in production of pollen. The mitochondria are also involved in respiration, and some of the male sterility seems to be aborting electron transport or disrupting the mitochondrial membrane that's associated with electron transport in the tissues that are involved in producing pollen. So that's the mitochondrial side. Now on the nuclear side, the restorers, most of the restorers, not all, but most of the restorers come from a very big family of nuclear genes that are involved in RNA editing. And so these So plants can contain as many as 400 or 600 of these genes in this gene family. And this gene family um, is involved, as I said, in RNA editing, and RNA manipulation is very important for controlling gene expression and the production of proteins. And so what these genes do is they regulate other genes by manipulating the transcripts that are produced. So if you imagine a protein that's made in the mitochondrion, then first of all, to make a protein, there has to be RNA made from the DNA, right? The transcript, the RNA transcript. And then the RNA has to be made into a protein. That's translation. These genes that are restorers, they manipulate the RNA transcript. And they can either edit it by actually changing the bases in the RNA transcript and then changing the protein, or they can change the probability that a transcript is actually made into a protein, the translation probability. So they have a very powerful effect on changing the actual proteins or the production of proteins. And that's called RNA editing or RNA um, interference, a variety of things that they do. And so the restorers are typically in this family of RNA editing genes, and so these that are known to be involved in mitochondrial gene expression. And so what they do, what the restorer genes seem to do, at least some of them from this family, is that when a mitochondrial genotype arises that makes a new protein that somehow is triggering, let's say, apoptosis, programmed cell death in a particular way, that the restorer genes are getting after the transcript, this new transcript, and manipulating it to prevent it from being expressed. So that's the arms race. And there's evidence that the restorer gene family evolves very rapidly. So people look at the rate of evolution of these restorer gene families, and there are particular sites in some of the restorer genes which evolve particularly fast. And those sites are actually the sites that are involved in binding to RNA. So if you imagine that the genes have to evolve to bind to new RNA transcripts to deal with a new cytoplasmic variant, then you would imagine that it's exactly the particular amino acids that are binding to the RNA transcripts that would have to evolve very quickly. And that's what we see is a very rapid evolution of the RNA binding amino acids in these RNA gene families. So that's one line of evidence for very rapid evolution. And there's some other lines of molecular evidence. Now, certainly there are other molecular mechanisms, but I think this just calls to mind and these RNA editing genes and the mitochondria, for the most part, they're involved in an incredibly cooperative gene network that has to do with respiration, programmed cell death, and for the most part, they're acting in concert in a very cooperative way. And so the fact that the mitochondria and the nuclear genes are in conflict over the production of pollen happens simultaneously with the fact that the mitochondrial genes and the nuclear genes actually have very cooperative, highly integrated gene networks and fitness interests for most of their interactions. And so conflict and cooperation tend to go hand in hand in the genome. And this is typical of the genome, that cooperative interests and conflicting interests are deeply intertwined. And it isn't one or the other. But what you're seeing is these gene families being involved in both cooperative and competitive interactions. And that leads to a very interesting shaping of the nature of genomes through evolutionary history and at the molecular level. And this goes back to my argument that you have to be very careful about engineering arguments when you look at gene regulatory networks, because gene regulatory networks are primarily shaped by the cooperative interests, and so the engineering viewpoint is okay, but there's these conflicting aspects as well as other aspects that really differ from human engineering, that really change the nature of genomic engineering compared to human notions of engineering. And a lot of the challenges in biology are to use principles of engineering, but to see the specialness of biology and to somehow see both at the same time. This is one of the great challenges. And again, why this topic is a particularly interesting topic. Um,
So does this also happen for chloroplasts?
I don't know of any evidence. I haven't really looked at the literature a lot over the last few years, but there really is no evidence that chloroplasts are widely involved. And that's an interesting question, why? And one possibility is that the mitochondria, because of their physiological role in programmed cell death and in respiration, can really target um, deterioration of the tissues involved in production of pollen in a fairly natural way given their physiology and genetics. Whereas for the um, chloroplasts, it might be not as easy to imagine the targeted um, attack of pollen in a way that's effective. That's a speculation, but it's consistent with what we see. That's a good question. Yeah, definitely.
Thanks.
Might you expect some collateral damage from having to like evolve against this sort of weapon the mitochondria has? Like if you're too effectively tamping down programmed cell death, then would you expect more incidents of cancer or something?
Well, that's a good question. That's one of my arguments is that when you see a conflict, even if it's resolved, you're getting an opposition of strong forces. And when you have an opposition of strong forces, misregulation is very easy. So I would think along the same lines as you. Of course, cancer in plants is a little bit different because of the nature of plant tissues. Um, but but misregulation would be something that I really think is one reason why you need to pay attention to these conflicts, even if they're not very common or mostly resolved, is that um, they're not engineered for robustness and smoothness of operation and minimizing error, but they're often engineered just to just to cope with this incredibly powerful conflict. And that really changes the nature of the forces involved and the engineering issues involved and the potential then for disease, which is one reason why my emphasis on where failure reveals design is often quite good. That when you see misregulation, disease and misregulation are often the same thing. When you see misregulation, it's often interesting to look at that and ask, what's going on? Is there something special about the design of the regulatory controls of this particular trait? Sometimes the answer is no. Disease happens, and all traits get misregulated. But, there's maybe particular misregulations and more common, more severe for these sorts of traits. That's my view. And so I think that's one reason why it's a very interesting to think about these things.
Cool.
All right. A few minutes.
Something I was thinking about. So I know in the past we kind of mentioned protein as a limiting factor. So if there's this arms race between them and they're developing different ways to counteract each other, so to speak, at what point does protein become a limiting factor in there?
Well, it could be. I don't know that we're coming up against that. And multicellular eukaryotic cells are a bit different from single-celled prokaryotes. Single-celled prokaryotes have to really be tuned for efficient reproduction because cellular growth rate and fitness are the same thing to some extent. But in a eukaryotic multicellular organism, fast cellular growth rate is generally cancer. So the tuning of growth rate and proteome limitation is quite different in a multicellular eukaryote from a single-celled prokaryote. There still, of course, is ultimately a limit. But, you know, one of the interesting things about eukaryotic cells is that they seem to be very sloppy. There's lots of, you know, there's lots of DNA, there's lots of genes that, you know, non-coding regions, there's regulation is sort of overwired, as I like to say. They just don't have that same efficiency thing. So I think the limitation and the conflict must come into play at some level, but it's a little harder to see with the same stringency that you would see in a prokaryote. Now, having said that, there's lots of prokaryotic conflict situations where that question would be appropriate for sure, like the toxins that bacteria produce, bacteriocins, for example, and restriction modification, you know, defense against viruses. How much of the bacterial cell can be devoted to defense? Some, obviously, with CRISPR and restriction modification enzymes, but a bacteria has to be fairly limited in its allocation of its genome to defense, because if it's devoting too much of its genome to defense, its genome is either big and it's going to slow down replication, or it has to trade off other functions that we don't see those trade-offs in multicellular eukaryotes. So I mentioned some of the other types of conflict situations, and I think that that's interesting. Obviously, if you look at host-parasite interactions, you can often see these arms races and multiple genotypes of pathogens and multiple resistance types, because generally speaking, conflicts and arms races come down to what I like to call recognition and polymorphism. The attackers are trying to evade recognition. In this case, you can think of recognition by the male sterile mitochondria as not having the new open reading frame protein that they produce recognized by the restorers that can RNA edit it, right? So it's escape from recognition, and on the attack side and on the defense side, it's recognition. And in pathogen defense, almost all of pathogen defense comes down to recognition, because the defenses that are used are often fairly generic. It's recognizing the attack, the attacker or the site of attack, and then bringing the defenses to bear that's so important. And so much of biology comes down to, I think of as shape and charge, right? Shape, charge, and binding. Often the recognition and polymorphism is manifest at the binding between different molecules. And so a lot of this arms race, you often see it playing out over at the molecular level in terms of recognition and escape and polymorphism. And then these sorts of polymorphisms over time and space often occur as well. And we see this in plant. Some of the best developed systems for studying this are in plant pathogens and the genetics of resistance to plant pathogens, where we see a lot of the same themes of spatial variation, turnover over time and space. Polymorphisms in a local patch maybe are restricted because there's been a recent epidemic where a particular pathogen is spread through and driven out all the other types and selected strongly for some resistance types and driven out some of the other resistance types. And so you get colonizations and extinctions over space and time. There's a lot of these recognition and polymorphism systems in bacteria. Bacteria are always trying to kill each other, and they use a variety of molecules that are broadly called bacteriocins, which are basically antibiotics that kill other bacteria. And all bacteria have many resistances to many different molecules that can kill them. And this is, again, played out at the various levels of binding and recognition. And so we see these tremendous polymorphisms where there are many, many different types of molecular shapes or variants of the attacking molecules. And on the defensive side, there are many variants of the resistance molecules. And again, with viral attack, bacteriophage, you see that if you look carefully, you'll see often a lot of variance in the bacteriophage, and on the host side, a lot of variance in the recognition of the different bacteriophage. And so you can imagine then and there being a lot of spatial dynamics playing out with regard to recognition and polymorphism. So this is a very broad theme that deals with many aspects of conflict. And with that, we can end. And the one thing I did not talk about that I hinted at was the reason why meiotic drive and a conflict within genomes is important for understanding Mendelian genetics. So you can think about that. That's your homework for the weekend. But I'll stay for a couple minutes if you want to ask questions, but if you want to go or have to go, feel free to leave, check out, and we'll see you next week. I'll post notes within the next day or two about the classes for next week. On Monday, of course, there's going to be a student-led presentation, and we've already had some of that on the website, and presumably we'll see some more details about maybe a suggested reading or two from that group, and then I'll post some things about class on Wednesday after I think a little bit about what I want to do next. So if you're leaving...
Sorry, working on the suggested reading
sorry?
list. We're working on the suggested reading list. We'll get it out
okay.
the next day or two.
I was just notifying everybody else that it's coming. Thank you. And if you have any questions, I'll stay here for a couple minutes. But again, if you want to leave, go ahead.